Very good morning to all. Am I audible?
Yes, ma'am, you're audible.
Yes, thank you. Thank you. Okay, let me formally welcome the session. Good morning to all the participants who are gathered here for a special lecture 6 series, which is on biomaterial as a drug delivery system, novel weapon against age-related diseases. This could be delivered by Dr. Berwin Singh. He is right now doing his postdoctoral fellowship at the Department of Foundational Science and Research in School of Dental Medicine, East Carolina University at Greenville. And to give an information about his education, he completed his MSc Biomedical Science at the Biomaterials University and went on to the MPhil Biomedical Technology at Sri Chitra Institute for Medical Science in the year of 2013. And he got his doctoral degree awarded by the year 2018 at Chonbuk National University at South Korea in biomedical science. And his research interest is focused on animal handling, cell culture, molecular biology, histology, and immunohistochemistry, and polymer synthesis, and finally material characterizations. And he has procured about 3 awards, notably BK21 plus Brian Korean Fellowship. And he is around 11 research articles published, both national and internationals. These few informations for our chief guest. I would like to invite Dr. Berwin Singh to deliver his speech on biomaterials as a drug delivery systems. So once again, I welcome you, sir. Now over to you, sir.
Thank you, ma'am. That was a wonderful and sweet introduction. So, good morning, everybody. It's a great honor to be in a virtual. We are able to communicate with each other, and thank you, everybody, for being here. Can everyone hear me? I'm just trying to check it out. Am I audible?
Yes, sir. Yes, sir. Audible, sir.
Cool. So, let me start with an interesting fact. How many of you know that a non-injured skin tissue has the ability to renew itself, that is, regenerate itself every 28 days? So next time, if your partner or if someone tells you that you're not the same person anymore, don't get angry. The truth is that you're not the same person how you were 28 days before. You know, so it's a fun fact. And with that, let me get into this presentation. In this presentation, you can expect to know, towards the end of this presentation, I expect you to know something about redox imbalance and how it's involved with age-associated disease, and something about smart polymers-based biomaterials and in vitro in a nano drug delivery system, and what is polyoxalate and copolyoxalate smart polymers, and how polyoxalate particles can be tailored to target the aging associated diseases. So the presentation flow is like a general introduction about aging, age-associated diseases, and then some of the topics would have some more specific introduction, and I'll talk more about polyoxalate, how it is synthesized, and how to make particles from the polyoxalate, and how polyoxalate particles can be used in cancer treatment and wound healing, and also in liver injury. So I'll touch, this is what you expect from this presentation today. Introduction to aging, associated disease and aging. In fact, like it's a known fact that aging is not a disease. It's a process. In terms of thermodynamics, it's a consequence of increase in entropy of the living system, which results in laws of molecular conformation. To put it in a simple words, we can say that as we age, the disorderliness of our cell and our body increases. So because of that, slowly we lose the ability to regenerate. Our system slowly, it starts to fail. And age-associated disease is a manifestation of these biomolecular events that results in loss of homeostasis, and it gives all sort of problems when you grow older and older. Reactive oxygen species, or chemically reactive molecules that contains oxygen and nitrogen oxide, nitric oxide, hydrogen peroxide, and there are other free radicals which are involved in this aging process. I'll talk more about hydrogen peroxide in this presentation. So moving on to the next slide. Hydrogen peroxide is the most stable reactive oxygen species. That is quite concerning because it can transfer through the membranes, it can travel from one cell to another, it can travel through the intracellular spaces. So it have a profound effect in our biological system. In a low to a moderate concentration of hydrogen peroxide, it helps as a secondary messenger, and it also helps in activation of certain enzymes. However, if the hydrogen peroxide concentration is shifted, if it is increasing, if it is left uncontrolled, it would impair cellular process and it would disrupt the cell viability as well. Abnormal intracellular hydrogen peroxide is a trademark of inflammation, cancer, and also in case of liver injury, myocardial infarction, and Alzheimer's, you name it. Even in case of COVID-19, what happens inside the lung tissue is that there's an abundant increase in the level of hydrogen peroxide within the lung. So majority of the inflammation, majority of the times, the culprit is reactive oxygen species and its imbalance. So as you can see here, the antioxidant and reactive oxygen species in a normal tissue are balanced, well balanced. There's a homeostasis which maintains this system. However, with age, the level of reactive oxygen species accumulation increases, and the antioxidants present naturally in our system, in our cell, can't mitigate the excessively produced reactive oxygen species. That leads to oxidative stress. So biomaterials are substances that has been engineered to interact with biological system for a medical purpose. It can be from an implantable material like stainless steel, or like a stent, or it comes in different forms. In general, we will classify biomaterials as organic and inorganic. In organic, you have polymers, and in inorganic systems, you have metals and ceramics. Biodegradable polymers or what breaks down into non-toxic by natural and form like it would release some natural products, including water, and it would not interfere with the metabolic pathway in our system. So smart polymers are a special case of biodegradable polymers. These days, we see a lot of smart products that exist in our daily day-to-day life. So smart polymers are kind of smart compared to that of conventionally available polymers. The smart polymers have the ability to respond to some stimuli that could be a physical or a chemical stimuli, like physical stimuli such as pH or temperature, and chemical stimuli could be either a redox responsiveness or an enzyme responsiveness. So these materials can be engineered in such a way that it can become a solid in the presence of certain pH, or it can break down in certain pH. We can engineer depending upon our need. Polymeric drug delivery utilizes the use of nanoparticles, and which are produced from the biodegradable polymers or biodegradable smart polymers. We can deliver a therapeutic agent which doesn't dissolve in water, and it can be released in a slow and a steady pace. So for drug delivery, any drug to be delivered into our system, it should be soluble in water. But sometimes some of the wonderful small molecules we have, it can't be dissolved in water. Say, for example, curcumin. We have turmeric, and turmeric consists of curcumin and other curcuminoids, which doesn't dissolve in water. So what we can do is that combine it with a polymer. We can encapsulate the drug inside a polymer, and we can deliver it intravenously or orally, so that the curcumin can be slowly released from this polymer in the matrix. As you can see here, this is just a pictographic representation how you see a polymeric nanoparticles, and we expect the water-insoluble drug molecule to be in the core, and this is the polymer layer, and this is how we formulate. I mean, this is what we expect when we engineer polymers for drug delivery applications. The polyoxalate and copolyoxalate polymers were first developed by Shalaby in the year 1970. It's with us for nearly 50 years now, And Ethicon company decided to make polyoxalate-based sutures because it's biodegradable and it doesn't interfere with our biological system. It breaks down after hydrolysis. It would give oxalic acid and diols, which doesn't interfere with our with kidney or liver. And the degradation rate of these polyoxalate or copolyoxalate can be modified depending upon the subunits we add to this polymer backbone. So this picture is a history of the polyoxalate. You can see 1970. It was the first time they published the molecular and crystal structure of polyethylene oxalate. And 1978 polyoxalate absorbable coatings were used in sutures. And by 2007, Dr. Murthy's group, they came up with this wonderful idea of using polyoxalate for chemiluminescence reaction in the presence of hydrogen peroxide, and they used it to sense the hydrogen peroxide. And that was actually the work was carried out by Dr. Lee, and that is my PhD supervisor. And later he used the polyoxalate nanoformulations for drug delivery. Moving on, this is the synthesis scheme of polyoxalate nano drug delivery system. So what we can do to synthesize copolyoxalate is that we can take water-insoluble small molecules that contain OH groups in it, and we can engineer the polymer in such a way that the naturally occurring small molecules can be incorporated in redox linker, and then it can be formed in a long polymer chain. The molecular weight of these polymers can be controlled. So we can make those polymers to degrade really quick, or we can design this polymer to degrade slowly. Thereby, it controls the rate at which the naturally occurring small molecules, like this natural small molecules which is present in the polymer backbone, to release either slowly or in a faster pace. So after synthesizing this polymer, what we do is there's one approach where we can make a polymeric particle, and these particle has the ability to degrade when the hydrogen peroxide concentration of the cell is higher. In any cell, there's a basal level of hydrogen peroxide concentration, which is quite normal. However, if you see in case of inflammation or cancer, the hydrogen peroxide concentration, the reactive oxygen species level, is very high than the normal physiological state. So when the particle goes to that interstitium, tumor interstitium, or the inflammatory tissue, it starts to degrade more rapidly than any other part of the body. The other way of making particle is polymeric self-assembly, self-assembled polymeric micelle. It involves the use of an amplifier, polyoxalate, and we can also incorporate another drug which can give an additive effect in our system, which could have an additive effect in the treatment. And it can also be applied for drug delivery. This is the first topic I'm going to present about. This is my first work I did during my PhD. So in this, I'm going to talk about how copolyoxalate particles is used in a novel cancer treatment modality. I don't need to define what is cancer. Most of us are well aware what is cancer. So there is a methodology to treat a cancer, which is photodynamic therapy. It uses a photosensitizer, a molecule which has the ability to convert the molecular oxygen into a triplet state, from triplet state to a singlet state. And these singlet oxygen are quite dangerous, and we use that to kill the cancer cell. The interesting thing about this is that the singlet oxygen is short-lived, and it can be produced only when we incident a laser light. And for an effective photodynamic therapy, we need a photosensitizer and a laser source. So when we shine the light on the tissue, which consists of this nanoparticle with this photosensitizer, in that local area, we can expect more cell death than any other, than the rest of the body. So it's more localized. But there's a limitation to this technique. The problem is the penetration depth of the laser. The laser has a penetration depth of maybe 1 millimeter. If your tumor is deep down your body, if it is inside a solid organ, we can't directly shine the laser light and expect this photodynamic therapy to work. Chemodynamic nanoparticle system is a photon-free system, so we don't need any light source for this system. The therapeutic system comprises of an amphiphilic molecule. We used puronic F127. An amphiphile or an amphiphilic molecule is like a lipid, like our lipid bilayer. It has one hydrophobic area and it has another hydrophilic area. And when we put a certain concentration of this amphiphile, it forms like a spherical micelle nanoparticle. So in this system, we use puronic, an amphiphile, a photosensitizer, and along with the polyoxalate polymer to form this self-assembly. So chemiluminescence, it's a process in which light, if you had seen the glow sticks, in that what reaction happens is a chemical luminescence. The diphenyl peroxalate, in the presence of hydrogen peroxide, it forms dioxane ethane dione, and this is a high energy intermediate. The high energy intermediate releases electrons, and if there's a dye which is present nearby, it would excite the dye, and the dye would release the luminescence, which we can see it in a visible way. That is chemiluminescence. So this is the schematic representation of the work. As I explained before, we used a puronic amphiphilic molecule and a polyoxalate polymer and a photosensitizer. If we combine it in an appropriate ratio, it would self-assemble and form a particle. So this particle would contain inside, it would contain a photosensitizer and the polyoxalate polymer. So when this particle goes to the cancer cell where the hydrogen peroxide concentration is higher, it would convert the molecular oxygen to singlet oxygen, thereby without any external laser light source, the cells would die. That's the concept. So this is a TEM image. We also checked the dynamic light scattering to know how much is the particle size, and it was around 250 nanometers. And this graph is a depiction of the activation of the particle and how the activated particle generates the singlet oxygen. So what we did is that without hydrogen peroxide, the di-9,10 dimethyl anthracene have a fluorescent intensity, and when we add hydrogen peroxide of 100 micromoles, it generates the singlet oxygen, and the singlet oxygen interferes with the fluorescent signal. So we know that singlet oxygen is generated in the presence of hydrogen peroxide. And this is cell viability. It shows like in the various concentration, like this is without, control cell, and this is just the polyoxalate polymer, and it didn't have any cell much of an effect. And as we can see here, just the micelle itself, it was not having much of a cell death. However, you can see here the particles which consist of photosensitizer and two different concentrations have a dose dependent cell death. This is a confocal fluorescence micrograph. The particles we tried to treat various times, and with time we can see the particle concentrated more inside the cell. This shows how the particles were uptaken by the cells, cancer cells, readily. A morphological assessment. I'll move on to the next slide because it would give you a more clear idea. This is for cytometric analysis of various formulation without treatment, with just the polyoxalate. You can see the apoptotic cells are not that much, and you can see in the presence of the particle, the concentration 100 microgram, there's a huge increase in apoptotic cell. So we also analyzed the procaspase expression, and we can see the normal level and what happens in 2 different concentration of polyoxalate particles. I want to mention something I didn't mention about it. You see the CA, what we did was that we tried to just use the polyoxalate formulation, I mean like micelle, and treated the cells. And we also used cinnamaldehyde. It's a small molecule which is present in the cinnamon. And what the cinnamon did, we pre-treated the cells with cinnamon of 100 micromoles. And why we did that is basically the hydrogen peroxide concentration is elevated in the cancer cell, but we added the cinnamaldehyde, we pre-treated the cell, and we increased the intracellular hydrogen peroxide concentration in cancer cell much further, so that when we treat this nano formulation, it would be much more effective. It would be like there will be higher generation of singlet oxygen within the cancer cell. That's why we also incorporated cinnamaldehyde. And findings, this study was the first report and a proof of concept of using polyoxalate-based, chemiluminescent-based, chemidynamic therapeutic system. And chemidynamic nanoparticle did induce singlet oxygen in the presence of hydrogen peroxide without any external laser light source. And polyoxalate chemidynamic nanoparticles, we propose that it holds a great clinical translational potential. And this is from the first page of my article, which was published by 2017. And yeah, this was my debut publication. This was my first publication that I ever published. And yeah, I can't be blowing my own horn, right? If I just say good about my work, what's the point? These are other works which were published later, and these were somewhat kind of using the similar principle. They also use chemiluminescence, and they also use polyoxalate, and they try to follow similar approaches. These 2 articles are proof. We just did cell-based assay, but these articles did a more descriptive study, as you can see towards the right. The study which was published in Chem was also the same, but they used the animal model, and they did show that there was a shrink in tumor size, and they did follow this similar methodology and similar concept. So it was pretty exciting when I saw these articles, and then it's like, wow, that's really proves my concept. So let me stop here. Do you have any questions so far? I would like to interact with you. You know, it's been how long is it? Do you have questions, guys?
Thank you, sir. Thank you for your wonderful lecture delivery. So,
No,
participants, any
I
doubt?
still have more to go. I am not done yet.
Clear your doubts now.
I'm just, I just
So one
wanted
question is
a
raised
short
from
break.
Sarita.
I just want to listen from
That
people.
question is regarding polyoxalate. If the cancer induced by certain chemical or a tumor antigen, for example, methylchlorothrin, is a polyoxalate react with the chemicals.
Can you please say that once again? Repeat the question one more time. Ma'am, can you please repeat the question one more time?
Sarita?
Ma'am?
Just raise your questions, ma'am. Sir is waiting.
Sir, my question is regarding to polyoxalate, sir. Polyoxalate, sir.
Okay.
Sir, suppose the tumor is caused by chemical. Is the polyoxalate react with that chemical, sir?
I can't say that for sure. It's a very good question because it depends. Normally it wouldn't react like polyoxalate or biocompatible polymers. When we check, that's why you can see in our biocompatibility analysis, like under normal circumstances, polyoxalate don't have any effect. You can see here, this is polyoxalate polymer alone in cell. It doesn't have any interference. But if you are saying if there's another chemical, maybe it could interact with those chemicals. But mostly polyoxalate or hydrogen peroxide responsive polymers, they don't react. They have very specific reactivity towards hydrogen peroxide than any other chemical components. I hope that explains your question.
Okay, sir. Thank you, sir. Sir, one more question, sir.
Sure.
Can you give the example of chemiluminescence?
Chemiluminescence.
An example.
Are you aware of the forensic technique where they would use the hydrogen peroxide ion, and they would, in crime scene investigation, they would use this hydrogen peroxide and I think horseradish peroxide. Like there's one reaction which involves iron. In blood we have iron, right? So like the crime scene investigation uses this technique to see where there's blood. They use similar methodology, similar chemiluminescence methodology.
Sarita?
Sir, ma'am? Ma'am?
Yes, ma'am.
Yeah, I actually, I really don't, that is the one way to look at it. If you could look up chemiluminescence, forensic, chemiluminescence in forensic investigation, in detection of blood, you can find it.
Okay, sir. Thank you, sir.
Read much in much detail in that.
Okay, sir. Sure, sir.
Okay, Sarita. Thank you. Kindly mute your mic. So other participants, kindly raise your questions and clear your doubts now.
Don't worry, I'll proceed with the rest of the presentation. Maybe if you guys have any questions, you can ask me later. I have some issue with the presentation. When I'm presenting in full screen, I don't have any access to this meeting window. I don't know why it's like that. All right, let me get back in track. But this is my second study on copolyoxalate nanoparticles, which was used for wound healing. Wound healing is a natural replacement of devitalized and missing cellular structure and tissue layers. This was one estimate by the UK. They said that wound management imposed an estimate of 5 billion pounds just in UK. This is to give you a perspective that we come across people who have wound like first degree or second degree burns. There are various ways we would get wounds. We can get hurt. It can be a bruise, or it can be a cut, or sometimes it's more dangerous in terms of burnt wounds. So it's a major problem, and as people age, their ability to heal naturally is impaired. And hydrogen peroxide, which is a clinically accepted biocide for killing bacteria, it's normally higher in wound. Even if you don't add hydrogen peroxide to your wound, we have this practice where if you get hurt, you use 3%, I guess, 3% hydrogen peroxide over the wound. It's a common practice, but there's a problem. When you add the hydrogen peroxide, it could affect the normal wound healing process. And what happens in wound healing, like you have an exudative phase and then a reabsorbative phase and a proliferative phase where the cells regenerate, and a repair phase. And in repair phase, if the repair is not completely proper, we would have some sort of callus and callus formation, and we can have a scar which could be very big. So this is one form of oxalate polymer. In this oxalate polymer, we have vanillyl alcohol. Vanillyl alcohol it's a natural small molecule from vanillin, or vanilla we have in ice cream, and it's one of the most famous flavors. And we use that vanillyl alcohol in our polymer backbone when we synthesized it. And with that, we prepared the nanoparticles, and we treated the nanoparticles in the wound, and we saw what happened during the process of 7 days. We used mice. We did a small excision wound on the back of the mouse, and we added the particle. We tested out what happened after in the presence and the absence of the nanoformulation. So when the idea is that in the presence of hydrogen peroxide, the vanillyl alcohol will be released at the site of the wound. Excuse me. So PVX particles or polyoxalate particles, this is the size. The size of the particles were around 250 nanometers, and this is how the particles would look like under a scanning electron microscope. And we studied the stability of those particles. The particles were found to be stable under physiological conditions. We tried in both physiological state and also in the presence of fetal bovine serum to show to mimic the biological environment. This shows that the particles are biocompatible. We used two types of cells here, and we found out the particles were quite compatible with those cells. And we also injected the particles in mice thigh and the muscle, thigh muscle, and we found out the polyoxalate nanoparticles didn't cause any inflammation or any damage to the tissue. This is commercially available PLGA nanoparticles. This is quite available. These polymers are FDA approved, but the lactic acid which is released from this polymer could induce inflammation. So we compared that. We know that it would induce inflammation, so we used that. We compared that with our oxalate nanoparticles. It shows the polyoxalate particles are quite superior to the PLGA particles. And this is a cell migration assay. We used 3 different groups: untreated particles 100 microgram, and particles of higher concentration 500 microgram. And we treated for various, we made a cut. We cultured the cells, we induced a cut in between, and we studied how long it took to close, like how long it took to close. And we can see here under lower concentration of particles, by the end of 12 hours, you can see there was a complete closure, which is in compared to untreated. You can see the wound didn't close. And you can see here in the presence of higher concentration of nanoparticles, you don't see a closure. This is quite important. I'll explain why in the next slide. You can see the excisional wound model. We made an excisional wound in mouse back, and we studied for 7 days. We did analyze those samples based on their own size, and this is what we found. This is hydrogen peroxide concentration in the wound. We took the wound tissue and we analyzed how much hydrogen peroxide is present in the wound tissue. This is just the skin, the basal level of hydrogen peroxide was present in the skin. And if we see the wound, the concentration of hydrogen peroxide was quite higher than the basal state. And if you see the 2 concentration, 2 milligrams and 5 milligrams, 2 milligram was able to scavenge the excessive hydrogen peroxide. The vanillyl alcohol was released, and the vanillyl alcohol scavenged the hydrogen peroxide. And in 5 milligrams, we can see there was higher scavenging of the higher hydrogen peroxide. There's a well-known saying which is, too much of anything is not good. So that is what we can see here. Yes, we need to scavenge the unnecessary amount of hydrogen peroxide, but we should make sure we don't overdo it. We don't scavenge the hydrogen peroxide to that extent where it would also affect the healing process. That's why you can see here in 5 milligrams, you don't see a complete wound closure. That is because the secondary messengers were interfered, because the hydrogen peroxide concentration was much lower, so it was interfering with other metabolic pathways. That was what we learned from this, and we saw this both in vitro and in vivo. So that was a bit interesting result. And this is the expression of hemoxygenase 1 and NRF2, and why this is important. Here's why it's important. So when there's like ROS, or high increase in hydrogen peroxide concentration, it would activate the superoxide dismutase, hemoxygenase 1. So these enzymes would reduce or keep the hydrogen peroxide in check. So if there is increase in NRF2 concentration, that means there will be an increase in hemoxygenase 1. So the wound healing process is happening. We found that hemoxygenase 1 and NRF expression downregulation was observed. So you can see here, this is just the wound, and you can see in higher concentration of the polyoxalate particles, the hemoxygenation, the expression is much lower. That's why we saw the wound healing was impaired. It's same with NRF2. This is image analysis of hematoxylin and eosin imaging. We quantified the epidermis and dermis. We did image analysis, and we found out the thickness of the epidermis and dermis, and also the collagen area. Collagen deposition in the wound signifies how good the wound is healed. If there is more collagen deposition, that means like wound healing is happening, and it is more good. You can see the skin and just wound without any particles. This is wound with free vanillin alcohol of 1 milligram, and this is wound with vanillin alcohol of 5 milligrams. And this is small molecule itself. There is no nanoparticles in this group. Both groups consist of just the small molecule, but here we used 3 different concentration of nanoparticles: 1 milligram, 2 milligram, 5 milligram, and we can see that like the collagen area is comparatively larger with increase in concentration of the nanoparticles. So findings. Reduction of hydrogen peroxide was observed in a dose-dependent manner in the presence of the nanoparticle formulation. PVX nanoparticles, that is polyoxalate nanoparticles, suppressed the generation of hydrogen peroxide in the excision wound. The downregulation of cytoprotective NRF2 and hemoxygenase 1 confirms effective regeneration. Hydrogen peroxide scavenging PVX nanoparticles offer a potential new approach for wound therapy. This was again published by 2017. The article, you can look it up if you are interested. Moving on to the final work. This is copolyoxalate particles for acute liver failure, and an acetyl P-aminophenol. That is, we call it as paracetamol in our place. It's quite common for if you have headache or if you have fever, you just to a pharmacy, you can purchase it, and it's pretty effective. However, there's a dangerous side to that drug. Abuse of this drug could cause a predictive drug-induced. If you are a person who consumes Tylenol more frequently, in paracetamol, if you are in a more frequent manner, it's not good. It would induce hepatotoxicity. It would lead to a massive production of reactive oxides and lipid peroxidation in liver. So if you're a person who consumes paracetamol more frequently, beware of this. Please try to avoid as much as possible. And also try not to consume too much in one time. At a time, don't just take it like a coffee or tea. It would kill you. So this is a synthesis scheme of POC or oxalate particles. So in this oxalate, copolyoxalate, what we use is this is curcumin from turmeric. We incorporated this along with the oxalate linker and prepared the particles, and we made particles out of it, polymers, and then we made the particles out of those polymers, and we tried to evaluate those polymers. These are all characterization data of the polymers, NMR and GPC to know the molecular weight of the polymers, and this is the fluorescence intensity of the polymer and just the small molecule itself. And we did check the intensity in the presence and absence of hydrogen peroxide, what happens when we add the hydrogen peroxide, and what happens to the polymer backbone. We did study that. We found out that when we add hydrogen peroxide, it can excite the curcumin, and it could increase the signal. So these are all particle characterization. Again, particle size, the particles were smooth under normal circumstances, but you can see without hydrogen peroxide, after 24 hours of incubation in PBS, it didn't have much effect. It's almost similar to how it was before. And in the presence of hydrogen peroxide, it started to break down and fall apart. And we also studied how the hydrogen peroxide increase would release the curcumin, like curcuminoids. So in the presence without hydrogen peroxide, the release was much lower. And if you see in the presence of hydrogen peroxide, the particle released more of curcumin. And this is to show that the dose-dependent release of activity of the polymers. So the schematic representation of what I did was the acetaminophen was given to induce liver injury, and after inducing liver injury, we treated the particle and we analyzed the liver under ultrasound, and we evaluated what happened after that. We sacrificed the animal and we took the liver and we studied how much was the molecular markers present in blood, and other minor studies were also carried out. This is to show how the particle did not have any bad effect. It was biocompatible. This is raw cell, this is macrophage, and the particles, even at a higher concentration of 200 microgram per ml, the particle didn't have any apoptotic or cell, it didn't induce any cell death. And we also tried to use an NiH3D3 cell, and we studied without add any hydrogen peroxide. You see, in the absence of hydrogen peroxide, these cells survived. It didn't have any effect, but when we treat hydrogen peroxide and give the particle started to degrade, and it started to release the small molecule, and it had a protective effect over the NiH3D3 cell. So you can see just hydrogen peroxide alone, it was killing the NiH3D3 cell. But when we add hydrogen peroxide and treat the particle, it protected this cell from dying. And this is another experiment to prove how, in the presence of hydrogen peroxide, the particles started to activate and reduce. You can see A is untreated cell, and E is higher concentration of particles, and it's compared to, if you see B, only hydrogen peroxide, the hydrogen peroxide concentration is higher, and we can see that from this. 10 microgram of particles and 25 microgram of particles reduced the intracellular hydrogen peroxide, and it scavenged the hydrogen peroxide. So the particles exhibited good biocompatibility, and it also proved to exhibit anti-inflammatory activity in vitro. And this is uptake, cellular uptake, untreated, just free small molecule of curcumin, and polyoxalate curcumin of 100 microgram after 2 hours, and polyoxalate particles of 100 microgram after 6 hours. So you can see curcumin of same concentration, it was not, the fluorescent signal intensity was not that much, because majority of the curcuminoids would have got degraded by the media. But in this case, the particles were initially accumulated on the cytoplasm, and with time, we can see the particles were localized in the cell. So this unstimulated raw cell means we didn't stimulate the cell, so there is no hydrogen peroxide. In the absence of hydrogen peroxide, it's quite similar to that of just curcumin alone. The activation of particles is quite lower. But if you see the stimulated raw cell, which consists of higher concentration of intracellular hydrogen peroxide, the particle readily degraded, and it started to release the small molecule to the cellular surrounding. This shows that polyoxalate particles were uptaken by macrophage, and in the presence of reactive oxygen species, the particle degrade more. We also studied the ALT, alanine transferase, the enzyme in liver. We took the blood and we analyzed how much ALT enzyme from the animal was released. We studied for controls versus acetaminophen treated and particle and acetaminophen treated group. As you can see here, the acetaminophen treated group treated with particle had a lower concentration of ALT, which signifies the therapy, which shows that the particle releases the curcuminoids in liver, and it subsidizes the reactive oxygen species in the liver. And this shows the biocompatibility, and the particle didn't have any effect on the lung, kidney, or heart. This is a tunnel assay. It shows that the polyoxalate particles reduce the apoptosis in AP treated acetaminophen treated group. It is just acetaminophen, and this is acetaminophen with particles. You don't see much of an apoptotic cell except for few. This is free small molecule of curcumin. We can see that it's not that water soluble, so it was not that effective in comparison to the polyoxalate, which consists of the curcuminoids. This is in vitro ultrasound imaging, and polyoxalate particle concentration increases the echogenicity, we found, because the polyoxalate particles have the ability to generate bubbles, CO2 bubbles. If you remember the schematic representation, like the particles, when it degrade, it forms carbon dioxide bubbles, and it also releases the curcumin. Let me go back to that slide. Yeah, you can see the particles in the presence of hydrogen peroxide. It hydrolyzes and it releases the curcumin, and it also releases carbon dioxide. So this carbon dioxide is more located in a local area, around the surface of the particle, so we can visually see that through ultrasound. That is what we try to study. Let's see, just the control and particles in the, this is acetaminophen liver and polyoxalate particles. We can see there's a huge signal increase compared to that of control. So in the acetaminophen induced liver injury, the polyoxalate particles exhibited more higher signal intensity. So findings: polyoxalate particles inhibited reactive oxygen species generation, and it protected the cell from hydrogen peroxide induced toxicity. In acetaminophen liver failure model, polyoxalate reduced the ALT level, and it enhanced the ultrasound contrast, so we were able to image it. It also exerted antioxidant and anti-apoptotic activity in liver. In summary, naturally occurring small molecules and their novel structures as a drug entity is still an active area of research. That means, rather than finding or looking for something from an artificial source or a synthetic source, we can look at small molecules which we already have and try to improvise how we can deliver this small molecule more effectively. You know, like, so it's still relevant. All the above discussed topic, we didn't find any new novel small molecules. So we are living amidst a pandemic. We're not trying to look for, at times we are trying to look for some simple remedies, like, using turmeric in our daily day-to-day life. we are using it and we are anticipating that would work, right? We are not looking for a vaccine right now, because it's a hard process. So we are looking for simple things we could do, like we make some concoctions with ginger or turmeric in warm milk. We tried to drink those and we believe it could work. So these drug delivery techniques using polyoxalate and copolyoxalate are similar. We use already existing well-known small molecules, and what we try to do is we incorporate it in covalently, incorporate in a polymer backbone, and we make a polymer and we try to deliver it. So polyoxalate and copolyoxalate small polymers are redox responsive polymers, which can be used in drug delivery. You can make it as a coating, or if you want to make a solid nanoparticle, you can make as a solid nanoparticle, or if you want to encapsulate in a micelle or self-assembly, you can do that as well. And you can play with it. You can literally play with it. So polyoxalate polymers are biocompatible, and it's biodegradable, and it's more superior to PLGA. I can say that. And polyoxalate particle formulation have tremendous capability in targeting aging associated diseases. So that's it. Questions? Before questions, give me a sec. I want to, acknowledge people who are involved. First, I would like to thank Ratnam College and Microbiology Department, the head of the department, Dr. P. Srinivasan, the department coordinators, teaching, non-teaching staffs, and students who were involved in organizing this special lecture. And thank you for inviting me. And I would like to give some special thanks to Dr. Nagendran Dharmalingam from Brown University, my former mentor, Dr. Dongwon Lee from Chonbuk National University. He was a great guide, and my parents, family, and friends. Last but not least, I would like to thank you all for your patience and coming here virtually, joining me to listen to my talk. I hope you understood something. And questions?
Thank you, sir. Thanks for your nice presentation. Sir, one more question from audience from Vaishnavi Prakash. His question is, can we use polyoxalate nanoparticle as a biodegradable and biocompatible drug delivery vehicle?
Yes, we can. The whole topic was trying to put all together and showing how we can use it as a biocompatible and polymeric drug delivery system. Yes, we can.
Okay, sir. Thank you, sir. Okay, now I call upon Ms. Krishma ma'am.
Yes, ma'am.
Yes, ma'am.
Okay. So, a very good morning to one and all. Is it audible?
Yes, ma'am.
Ma'am.
We can hear you.
So, I hope you may all have enjoyed this session, and it is my great privilege to deliver a vote of thanks for this session. So, I, on behalf of Department of Microbiology, Ratnam College of Arts and Science, wish to thank Dr. R. Manikyam, CEO of Ratnam College of Arts and Science, for his support and guidance for conducting this program. Next, I may like to express our sincere gratitude towards Dr. R. Muralidharan, Principal of Ratnam College of Arts and Science, for encouraging us to organize such events. Thank you, sir. And a big thank you to Dr. Berwin Singh, our chief guest, for the excellent presentation on the topic Biomaterial as drug delivery system and novel weapon against age-related diseases. And sir, it was an amazing session, and we all have got an idea of the role of biomaterial and its effectiveness in drug delivery and against age-related diseases as well. And sir, it is. Thank you so much for sharing your opinion and finding with us. And it was our great pleasure to have you here, sir. Thank you so much. Next, I must mention the organizer of this event, Dr. P. Srinivasan, the head of the Department of Microbiology. Thank you, sir, for organizing such a wonderful event. And I'm very grateful to Dr. Rajalakshmi, Assistant Professor of Microbiology, Ratnam College of Arts and Science, for coordinating the event in an excellent manner. Thank you, ma'am. Next, we would like to acknowledge our gratitude towards all the participants who have been present here for your cooperation and your participation. You people have made this program a big success. Thank you all.
Thank you, ma'am. Thank you, Krishma, ma'am. So participants, kindly stay on online. Within 5 minutes, I will share a feedback link. Sir, thank you very much, sir.
Thank you.
Okay, thank you.